Hello, hello. I am so happy to be back with you guys again in this new edition. We are about to start chapter 4 of The Myth of Normal, Dr. Gabor Mate's most recent book. And today we have a very interesting topic because the topic of genes and experience has been a very controversial topic. And it is my honor as a former scientist to further give us a look of what Dr. Gabor proposes and how we can actually make this very easy to understand and really connect with the message behind how our genes get affected by our experiences. I say hi to everyone that is joining this transmission live on YouTube, live on Instagram, and just, tell me your comments, tell me your questions. I'm more than happy to answer to the best of my capacities. And let's start with this reading. Chapter 4. Everything I'm surrounded by. Dispatches from the new science. So much of what makes people either well or not is not coming from within themselves. It's coming from their circumstances. It makes me think much more about social justice and the bigger issues that go beyond individuals. In 2009, Dr. Elizabeth Blackburn shared the Nobel Prize in Physiology or Medicine for her work in telomeres, minuscule DNA structures at the end of chromosomes. Not unlikely, the plastic aglets placed at the end of shoelaces to keep them from fraying, these tiny sheets help protect chromosomal integrity. Good thing too, since as chromosomes unravel, so do we. Tracking the length and stability of telomeres throughout the lifespan, it turns out, can tell us a great deal about our health and longevity. Okay, you wouldn't think to look at them, but what has been discovered about these tiny biological structures also has huge social implications. How so? How come something such as tiny as a telomere, as a part of the structure of a DNA, can have social implications? Have you asked yourself that? Amazingly, she found that factors such as poverty, racism, and urban blight can directly impact our genetic and molecular functioning. As a psychologist, Elisa Epel, who is Dr. Blackburn's research collaborator and co-author of the best-selling volume, The Telomere Effect, a revolutionary approach to living younger, healthier, longer, told me in an interview, these effects are not small. The neuroscientist Candace Lewis, whose own research is in epigenetics, the growing field that investigates the impact of life experience on the activity on our genes, sees things the same way, quote unquote. More and more, the science is demonstrating this holistic model of who we are, she told me. It's more than just what's enclosed in my skin, it's everything I am surrounded by. As Dr. Lewis has peered at molecules and strands of DNA, she too has found herself lifting her gaze to the whole person and from the individual to broader social issues. As a specialist in the complexity of brain and behavior, I know it's not just brain and behavior, said the former Fulbright scholar. One of the biggest take-home messages from my work is how malleable we are as an organism, how responsive to environmental cues throughout the lifespan, how responsive to environmental cues we are to our lifespan. The dominant assumption in our culture is that genetic inheritance determines the better part of our destiny. Who we are, what we suffer from, and what we're capable of. In 2000, at a White House briefing, Bill Clinton proclaimed the findings of the Human Genome Project, the most wondrous map ever produced by humankind, adding that today we're learning the language in which God created life. The new science, the soon-to-be expressed and predicted, will revolutionize diagnosis, prevention, and treatment of most, if not human diseases, leading to cures for conditions such as Alzheimer's, Parkinson's, and cancer by attacking their genetic roots. And I just want to mention a brief note that is right here below. There are a few diseases that are determined purely by genes, such as Huntington's, and one that runs in my family, muscular dystrophy. If one has the gene, one is almost 100% certain to get the disease. Such conditions are exceedingly rare. There is, for example, a gene breast for cancer, but only about 7% of women with the disease have the gene. And far from all the ones with the gene will necessarily get the disease, though the risk for sure is significantly elevated. This is to say that there is this belief that we are the effect of our genetics. And this is not true. What Gabor is proposing here is that we are the experience. Our genes are actually affected by the experience of our lives, the experience of our situations, and how we're interpreting and giving meaning to situations. However, there is a percentage, a very low percentage of gene diseases, that are really determined purely by genes, okay? 2 decades later, we know that little of the sort has happened, and for good reason. Genes are not, in fact, life's language. And more than a scrambled alphabet or a randomly arranged dictionary is a Shakespeare play, or a musical scale is equivalent to a John Coltrane solo. For letters or words to become language, they must be arranged, enunciated, inflected, punctuated with pauses, emphasized or softened. Like all building blocks, genes help make up the language of existence, but it is through the workings of epigenetics that they are activated, accented, or quieted. The mechanisms of epigenetics include, among myriad others, adding certain molecules to DNA sequences so as to change gene function, modifying the numbers of receptors for certain messengers, chemicals, and influencing the interactions between genes. Experience, in other words, determines how our genetic potential expresses itself in the end. This is what the field of epigenetics, meaning on top of genes, is all about. Epigenetic processes act on chromosomes, delivering and translating messages from the environment that tell the genes what to do. All this takes place without in any way altering the genes themselves. As the BBC's Martha Henriques explained, epigenetics offers a way of adapting to changing conditions without inflicting a more permanent shift in our genome. It isn't that genes don't matter. They certainly do, says Gabor. Only that they cannot dictate even the simplest behaviors, let alone account for most illnesses or address possible cures for them. Far from being the autonomous arbiters of our destinies, genes answer to their environment. Without environmental signals, they couldn't function. In fact, life for us would be impossible if not for the epigenetic mechanism that turn genes on or off in response to signals from within and from outside the body. Epigenetics revamps our understanding of human development from embryo to adult, and even how our species got to be here. I spoke with one of our foremost research in the field, Dr. Mossi Sif at McGill University, my previous university, storied medical school. Evolutionary theory is a difficult one to change because, it became almost a religion of science, he said. And any questioning of it seems to be a heretical question of the whole system, which obviously it isn't. Epigenetics doesn't deny evolution. Epigenetics is a part of evolution. I love this. but it demands a new look at how evolution works. The new biology improves upon the standard Darwinian view of spontaneous mutations and random selection as the motors of species adaptations. It demonstrates the circumstances themselves can shape how genes adjust to the environment. Why do you think this information is important for us? It's important for us to understand. Because modern science for decades has told us that we are the effects of our genes. This is not the case. The environment is. So if we are able to give meaning to how we process situations and how we deal with experiences, what do you think is going to happen with your genes? Said another way, our lives are what happens when life acts upon life. Dr. Sif and his team in Montreal performed one of the most cited epigenetic studies with major implications for how we view development, behavior, and health. Working with laboratory rats, they examined the effects of the mother's interactions with the infant in the first days after birth and how the offspring for the rest of their lives respond to stress, whether appropriately and confidently or with anxiety and overreactivity. The focus was the HPA axis, the stress-regulating feedback loop between the hypothalamus and the pituitary and adrenal gland. In particular, the researchers looked at the receptor molecules in the brain whose task is to modulate stress, which is to say, to ensure the appropriate behavior when stress is present. Creatures with poorly self-regulated stress reactions will be more anxious, less capable of confronting ordinarily environmental challenges, and overstressed, even under normal circumstances. The study showed that quality of early maternal I'm going to say it again. The study showed that quality of early maternal care to have a causal impact on the offspring's brain's biochemical capacity to respond to stress in a heavy way into adulthood. Key epigenetic markers, the way certain genes express themselves, were different in the brains of rats who had received either more or less nurturing contact from their mothers. Strikingly, the offspring in turn passed onto their own infant the type of mothering they had been given. Sif and his colleagues have also shown that the quality of maternal care affects the receptor activity for estrogen, a key female hormone, in daughters, with ramifications for mothering patterns down the generations. Isn't that interesting? Though ingenious manipulations on the rat population studied, unconceivable in human research, both the physiological and behavioral effects of early nurture patterns were found to be non-genetic. That is, non-transmitted through the so-called genetic code, which remained unchanged. Rather, they were epigenetic. So, the genes were not changed. They are epigenetic, in other words, determined by how the various kinds of maternal nurturing influence genes' activity. The experience of maternal care influenced genes' activity in the offspring's brain. The specific maternal behavior tracked by these researchers was how lovingly the moms groomed or licked their infants. Okay, but these are rodents in the lab. I was just thinking about that. You might find yourself saying, what do these findings mean for people in the real world? A reasonable question to which nature provided an eloquent answer in the form of a devastating ice storm in January 1998 in the same province in Quebec. No less, where Dr. Siv and his team did their work. Considered one of Canada's worst ever natural disasters, the storm left many Quebecers without heat or electricity. The more objective stress that pregnant women had to live through during those trying days, as in concrete, measurable factors like darkness, cold, and home damage, the more their kids' physiology was marked by that adversity, even near poverty. The participants were of a similar socioeconomic, cultural, and ethnic background and lived in the same suburban area. Over the years of the tracking of the children, Suzanne King, a professor of psychiatry at McGill University, said, "We found that the objective stress explained how kids varied one from another in a whole host of things: language, BMI, which is body mass index, and obesity, insulin secretion, their immune system. Even IQ was affected. We also saw increased asthma," Dr. Sif added, "as well as increased inflammatory genes and immune genes that are connected with autoimmunity. I should emphasize that mothers aren't alone in transmitting chronic disturbances of the body's stress apparatus to their young. In one experiment, healthy male mice were vexed by a series of stressors: frequent K changes, constant light or white nose, exposure to fox odor, being restrained in a small tube, and so on. Different stressors. They were then mated with non-stressed females who provided their pups with perfectly good mothering. Their young showed impaired stress response behaviors and blunted stress hormone patterns. In other words, despite the mother's best efforts, the fathers had transmitted the disturbing effects to their sperm. In humans, paternal stress early in child's life can also have long-term effects into adolescence at least. Adversely, among both mothers and fathers bear reliable linkages to the epigenetic profiles of the children, a group of researchers concluded. Socioeconomic circumstances, too, can alter epigenome, the web of epigenetic influences on genes. Indefatigable, Dr. Sif teamed with scientists from Canada and the US to study the epigenetic working of a broad range of genes in blood samples of middle-aged British males. The study subject had begun life at the opposite ends of the wealth to poverty spectrum, some poor and some others rich. Gene expression in those who were born well off was markedly different from that observed in their counterparts who grew up disadvantaged. Another study observed higher rates of inflammation in African Americans more than in Caucasians, an epigenetic effect that remained even when comparing those of the same socioeconomic level. We found that experiences with racism and discrimination accounted for more than 50% of the Black-white difference in the activity of genes that increase inflammation, wrote the lead author, Dr. April Thames, in an article titled "Racism Shortens Lives and Hurts Health of Black and by Promoting Genes That Lead to Inflammation and Illness." Much like gene expression, telomeres manifest the vagaries of fate and history, class and race, stress and trauma. How? At birth, telomeres have many units. The DNA base pairs, which they are constituted by old age, far fewer. We start out with about 10,000 when we are a baby, and when we get down to 4,000 when we die. Elisa Eppel told me, every time a cell in our bodies divides, telomeres shorten. When they get too short, their whole cells dies, or many deteriorate and become dysfunctional. As they shrink, immune function is impaired, inflammation rises, and we fall more prone to illness. Telomeres have been called cellular clocks in that they are a measure of biological rather than chronological age. Two people, even identical twins, could have the same age as computed in years, months, weeks, and days, yet one may be biological older than the other depending on how much stress, adversity, trauma they have endured. That's because stress shortens telomeres. Doctors should take special heed. The telomeres of medical residents suffer greatly attrition than those of other young adults in their age group. One of Dr. Eppel's studies found that caregiving mothers of a chronically ill children had shorter telomeres than their counterparts of the same age. This biological age deficit was proportionate to the number of years of caregiving and the degree of stress as perceived by the moms. Similar results were seen in caregivers of people with dementia, shortened telomeres and impaired immunity, reinforcing the idea that chronic psychological stress has a negative impact on immune cell function and may accelerate their aging. In other words, stress ages our chromosomes and therefore ages us. Just as poverty and racism affect epigenetic functioning, so do these factors also shorten telomeres and therefore lives. This sobering language was brought home vividly by a study of Black American men in 2014. Her findings literally suggest that racism makes people old. The author commented, the same holds were true for women. As part of the US-based study of a woman's health across the nation, the telomeres of Black and white middle-aged women were compared. The results were shocking. Black women were found on average to be over 7 years more biological age than their white counterparts, consistent with higher rates of poverty, stress, hypertension, obesity, and related health conditions. As Dr. Eppel told me, the effects of our socioeconomic environment are visible within ourselves if one knows what to look for. The neighborhood deprivation, the crime, the income of the zip code, she said, all of that is associated with aging of the cells. That is, to me, one of the biggest demonstrations that our health is outside of our body. Dr. Sip spoke in similar tones. For a century, we've been obsessed with chemical changes, thinking anything that is chemical is true, and anything that is not chemical is not true. What epigenetics taught us is that social changes are really not different than chemical changes. The one is manifested in the other. Fortunately, the DORA environmental effects swings both ways. It turns out that experiences that build stress resilience can lengthen our telomeres, even in the face of illness or adversity. This has been shown by the work of Dr. Eppel and colleagues with meditators, by Dr. Jim Brody's work with the deprived Black American teenagers, and in other research of men with prostate cancer. This can be a recurring theme as we proceed, the seemingly bad news giving way to something empowering if we approach it wisely. By learning about the impacts of adversity, we can also find pathways towards healing. And I just love the fact that he ends up in this very high note, because it's important. First of all, now we understand that our genes are affected by our experiences, and this is what the epigenetics field intends to describe. Second of all, now that we understand that stressors, life circumstances, trauma, and so forth are what is causing a specific gene in us to lighten up and to express in a certain way, now we can know, and now we know, that if we build stress resilience, then the opposite process is going to occur. In this case, the process of gene expression is going to become healthy and durable and resilient. So when he says here that we can learn about the impacts of adversity, in that sense that if you are going through an adverse experience, an adverse situation at this moment, if you're experiencing depression and anxiety, high chronic stress, please know that you can find pathways towards healing. That building stress resilience is the pathway so that your actually body can embody this stress resilience, giving you health, joy, and motivation to move on in life. This is the end of chapter 4, everyone. I am very happy to keep on reading what Dr. Gabor Maté is sharing with us. If you have any comments or questions, please let me know. If you want me to provide other sort of content as well related to his work in compassionate inquiry and his work in general, also please let me know in the comments. I wish you a beautiful weekend and see you guys next Friday. Bye-bye.